Anybody remember what the equation for cell respiration is? Glucose is in there, C6H12O6. Oh,
plus oxygen.
Oxygen, there we go. Then that gives us CO2, H2O, and ATP. Almost running out of room there, but we fit it in. So our whole job is to make ATP. We really absolutely need it. We would die without it. We can't store the stuff, so we have to make it as we go. So definitely in biology, one of those important molecules that we have to have. So, we review where we were yesterday. Like I said, this is probably one of the most abstract things that we do during the year is cell respiration, and then next chapter photosynthesis. But I've been getting you kind of ready for it the last couple days, and if you followed me so far, then you should be okay today. Because some of the highlights from yesterday, if you understand, and I'll review it now, that what we really want from our food molecule or our glucose molecule is the H's. Because it's the H's that we're going to use in the end to make kinetic energy, to make ATP. Because, well, this reaction here, P plus ADP equals ATP, that's an endergonic reaction. You got to put energy into it. It's not free. You can't just do it for free. You have to have energy for it. So we're going to use the H's in the end for that energy, the electrons, protons. So even though there's carbons and there's oxygen, we're going to get those out of there, and we're just going to go with the H's. But meanwhile, what are we going to do with the C's and the O's? C plus O equals carbon dioxide. Yeah, we're going to turn the C's and the O's into carbon dioxide and get rid of it, but we're going to keep the H's and send it to this part of the mitochondria and the inner part of the membrane. And we're going to do this. I drew this out a little bit to give you a kind of a sneak peek yesterday. You have your H's in the end, and you send the proton part up to one area, the electron part to another area. You separate them. Now you've created a concentration gradient, or you can say opposites attract type of gradient. And they want to get to each other, but you put a barrier in between them, and then put an enzyme here in the middle where that's the only way they can get to one another or flow down their concentration gradient. When they flow, they'll go past this enzyme, and it's, remember, the world's smallest turbine. It literally will spin, and I did a little spin here because I like to spin. It will spin that enzyme and give energy, kinetic energy, to do this phosphorylation reaction, putting the P's, excuse me, onto ADP to make ATP. So if you followed all of that so far, today's going to be easy, right? So yes, it's a little abstract, a little complex, but that's all right. Cell respiration comes in a few different parts. I gave you a preview of that yesterday as well. We're going to go through the parts, look at the highlights and why it is and what it contributes to this final ending piece. So let's start with part one. Let's set the location first. So we're actually not in the mitochondria yet. We're in the cytosol, which is the liquid part of the cytoplasm, the juice. So we're there. We are anaerobic. What does that mean? Without oxygen, this part does not require oxygen. The oxygen you breathe goes to the mitochondria.
All right. And in order to get this started, you have to pay. So it costs 2 ATP to start. Got to spend a little money to make a little money. So there's our setting. And the name of this process is called glycolysis. Which, if you know your Latin or your Greek, it kind of tells you what is going on. You have the word or the prefix gly. That means sugar. So our sugar in question here is glucose. That's the model we're using. And then the word lysis, or should I say the suffix lysis, is splitting. So that whole word tells you exactly what's going to go on. You're going to do some sugar splitting because we're starting with glucose and we're going to catabolize the glucose to get the H's off of it. So every time, well, you're going to change the molecule, you're going to change it around, pull some H's off. Eventually you'll be pulling C's and O's off of it, but you get rid of those. But you keep the H's, and so in the beginning, this is our last time that we can actually recognize glucose because we're about to start changing the molecule around and start splitting it up into pieces and taking stuff off of it as we go. So that costs a little bit of money in the beginning. So I'm just going to draw the 6 carbons that represent glucose. 1, 2, 3, 4, 5, 6. There they are. I know, yeah, there's H's and O's on there too, but I'm just going to draw the C's for now. So if we had to put a couple ATP into this, that means we had to phosphorylate this. So I'm going to put those on the end, put the P's on. We did that the other day. We even phosphorylated our sodium-potassium pump the other day. So it's just like that. You put the phosphorus group on there. It came from ATP. A kinase enzyme would phosphorylate things. And when you do that, you destabilize the molecule and you give it energy to do stuff, to do work. Like, in this instance, to break the bonds. So we're going to go right down the middle here. That's where we're going to split. That's a good, easy way to show that. And then we're going to end up with this and this. 2 3-carbon molecules. Nice and easy. Now what I just showed you, yeah, that looks easy, but in reality, if you go in your book and you check it out, it's actually a little more complex. There's about 10 different steps that are in between here. I'm not going to draw them out or even worry about them. If you want to see them, go ahead, open up your book, take a look at them. There's diagrams and things, but we're not going to worry about that at this point. So there's 10 steps in between here, even though I made it look really nice and easy. Every time we change the molecules around, it looks like a different structure. It's usually given a name. You can find those names in the book if you really want. I usually ignore those unless it's an important name of something we should know and it's probably going to show up again. So in this case, I'm going to give you a name for these because I want you to know the name for these. So each of these are called pyruvate. And, well, 2 pyruvates. I'm giving you that name because that name is going to show up again later on in the chapter. And might even show up later on in the year. So that's one particular name I want you to know. Otherwise, all these in-between steps, don't worry about those. Now in yesterday's notes also, remember we had some other players. They were the electron carriers or the H carriers. I acted like that yesterday. If I have an H, I know I'm not going to give it, I reduced him yesterday. But maybe I'll reduce Chloe. There we go. There you go. H. So there's those players in this too. They're waiting around in the wings. Every time an H comes off of there, it grabs it. So NAD is somewhere around here. And we did actually get maybe just a few H's from here. So let's see here. I'll put that one over here. So if an H comes off, it joins with NAD. Notice how I'm going backwards in my drawing. And then becomes NADH. And it's going to do like I just did. It's going to shuttle that hydrogen or that electron to the mitochondria. And it's going to give it something, or give it to something to get into the mitochondria, and then come back for more. So we do have that molecule waiting around. We also talked about FAD yesterday. There are no FADs here, but they are in the mitochondria and are going to help out there. We are also going to make 4 ATP off of this. Some of the reactions that are made, ADP gets phosphorylated, and we make 4 of those, even from just doing all of these steps of these chemical reactions in here. We make 4, but however, it cost us 2 to get started. We make 4, so how many do we net at this point? 2. So it's like paying your paycheck. You gross so much, but, well, Uncle Sam takes out what he wants, and then they give you your net. And you're like, what? They took all that out. I could have used that. Welcome to being a good American citizen, paying taxes. All right, now we have a choice. 2 choices, actually. Choice 1 and choice 2. Choice 1 and choice 2. All right, we're going to take the pyruvate and continue with what we're doing. Because we're still taking the molecules apart, trying to get the H's from them. But we have some choices. If everything is in good shape, you've got good blood vessels delivering good oxygen to the mitochondria, then sure, we're going to send the pyruvate to the mitochondria. But we'll put if O2 is present. And if you're just doing regular everyday activities, we're probably okay. However, maybe you're getting a little more active than you are normally. So, well, maybe you don't quite have enough blood vessels to deliver enough oxygen to your muscles in an efficient manner because you are not quite as in shape as you thought. Like somebody trying to run a marathon and they're not quite ready, or somebody who just got off the couch after all winter sitting around and now it's time for track season. Now you got to start running. They don't quite have the delivery of oxygen that we might need. So if oxygen is not adequate, we can also go to choice number 2. We're going to stay in the cytosol. And we're going to do something That's not going to feel good in a couple days. Well, in maybe one day, like 24 hours later, kicks in. We're going to do something called substrate level phosphorylation. There we go. I can hardly get it all written in there. Substrate level phosphorylation, meaning we're going to use our pyruvates to generate ATP right there in the cytosol. However, this is a little bit different process than what I'm talking about on the board over there, where we use oxygen and we use the inner mitochondria. In this case, we build up lactic acid. And if you've ever felt that, you know it's not very fun. It makes just a little bit of ATP. Not very efficient. Makes a lot of lactic acid, tears your muscles down. You're not getting enough oxygen, and then in about a day you're going to really have that kick or that pain kick in. It's going to last for a couple days. And then things will heal. That's the good thing. You'll heal. The lactic acid will be carried out of where it's sitting in your muscles. It'll actually go back to the liver and it'll be transformed back to pyruvate again. And maybe this time it can go through the right steps. This is the good one. Ooh, this is the bad. Do a little sad face on that one. That one makes you feel bad. Bacteria do something similar. However, in their process, it's called fermentation, and they make alcohol when they do it. Good thing you don't make alcohol, you make lactic acid. Or you'd be drunk just from trying to work out when you're not in shape. Woo. All right, but with our model, we're going to go and do the good one. We're going to go to the mitochondria. So now I'm on page 2, step 2. Any questions on step 1 so far? All right. For step 2. That was all step 1. Yeah. All right, so in step 2, this one is actually, to me, it's kind of like a half step, but your book puts it into a full step. So this would be like step 1.5 to me, but we're going to call it step 2 because that's what your book does. We're going to get pyruvate ready or change pyruvate. Change pyruvate and then send it into the mitochondria. So this is just a short little step here. I'm going to make it a little longer though, just for you, because there's some good stuff in this one. So let's just take one of those molecules we just made. Just take one. Yeah, I know we made 2, but let's just take one. It'll be easier that way. So here's our pyruvate. We're going to modify it a little bit to get it ready to go to the mitochondria in the next cycle. And we continue to catabolize this molecule because we want to get H's. So one of the things we do is add a carboxyl group to it. COOH, recognize that? Add the carboxyl. Oh, but look what I am looking at right there. I see an H. Who loves H's? Who's sitting nearby and wants that H? There's an NAD. It sees that H. And that gives me NADH. So NAD actually steals that H off of that carboxyl group, and it's going to take it, of course, to the mitochondria. Always to the mitochondria. So now, if I follow my molecule along, now I have just an OO on the end on that one. OO negative. And it turns out that that makes that really unstable. And while I'm not going to test you on this little reaction right here, I wanted you to see this reaction. Number one, because here's the carboxyl group coming back to haunt us again. Number two, you can see an example of NAD grabbing an H. That's always good. And number three, you can see what happens right here. Since this is unstable, we're going to pull that C off of there. And we're going to have C plus O, and that's going to equal CO2. I wanted you to see how a CO2 molecule can be made. After all, if I go back to our beginning, isn't CO2 one of the waste products that we're supposed to be generating? This is where we're going to start generating some CO2. So while I will not test you on this little area here, that little reaction, I wanted you to see that. Now you know. Why do I have to keep breathing out all the time? Now you know. It's that little reaction right there. Plus, there's going to be more in our next cycle in part 3 as well. You're going to generate some CO2s. Because we're tearing this molecule apart. You're going to get C's and you're going to get O's combined, get CO2. All right, this molecule though, I want you to know. So we've got a 2-carbon left. We took one off. Took that little guy off there. 2-carbon molecule, we're going to combine it with something called coenzyme A. So now it looks like CC CoA. You can just use CoA for short. This is the molecule that's going to go to the mitochondria. Now it's ready. Or fat. What? Who are you calling fat here? Well, your body can make a decision here. Should we send this to the mitochondria to make ATP? Or, you know what? It's just as easy to make that into fatty acids. Fat. So this is where your body decides, are we going to need to make ATP, or should we make this and store it as fat? What do you think? Well, if you're active and you need ATP, it will use it for ATP. More so than the other one, otherwise vice versa. So there's the decision. However, lucky all of you, we're going to go to the mitochondria with this. So that's our kind of a in-between step. We just modified pyruvate a little bit, created a little carbon dioxide, some H, and a molecule that's ready to go on or become fat. So let's go to step 3. Step 3, Krebs cycle, or sometimes called the citric acid cycle. Krebs is capitalized. It is named after someone. I suppose it just gets out of hand that everybody wants something named after them, and using people's names sometimes gets difficult. And then if you have some people claim, well, I discovered it first, no, I did. Then some people call it Krebs, some might call it Johnson cycle, and well, then nobody knows what we're talking about. So it's just sometimes better to have the real name, or just a name everybody can agree on. Alright, so citric acid cycle or Krebs cycle. We are in the mitochondria now. By the time we're done today, we will have written mitochondria 20 times. Now, not that much. So, we have our 2-carbon molecule. The CoA is shed. So I'm just going to go back to doing my CC. The name of your kitty? CC, right there. Alright, then I'm going to make a big cycle thing, because that's what it is. It's a cycle. It's a cycle that these different molecules go through, how we rearrange it, tear it apart, do different stuff. All hopefully trying to get H's off of this stuff. So this is basically a cycle of reactions. I'm going to use the word harvest because it's fall out there. To harvest hydrogens or electrons, whichever one you want to call it. Some books strictly say electrons. I like to include the hydrogens because electrons don't travel alone. Now in any reaction, if you're going to join a cycle or join a circle, you have to like get into the circle, right? If you have a circle of kindergartners and they're all holding hands, how are you going to get into that circle? Somebody's going to have to let go and grab onto the new hands of the new people coming into the circle. Well, there's some molecules coming off of this cycle. It's a 4-carbon molecule, and its name is citrate. I will not test you on that, but I just want you to know that that's where we get the word citric acid cycle from. However, knowing that there's a 4-carbon molecule coming off that, you should know 4-carbon molecule joins hands with this 2-carbon molecule and gives us, again, we're back to a 6-carbon molecule. 1, 2, 3, 4, 5, and 6. I don't know the name of it. I don't care the name of that one. You want to look it up? You can. There's a big picture in your book. It probably takes up a whole page. It shows the Krebs cycle and actually shows you all the different reactions going around this thing. But the main idea is that as we take this and we rearrange this, H's come off here. So I'll put some H's coming off. Electrons, H's. And who's going to want to grab them? NAD. So NAD is over here waiting. NAD's waiting. Oh, and in this one, FAD's waiting too. FAD, and that one can take 2 H's. Those are like your vitamins, remember? Get your good vitamins so that you can grab as many H's as you can to make good ATP. So these guys will shuttle these electrons to the inner part of the mitochondria. Oh, there's that word again, mitochondria. Can't fit it all on one line. So there's our shuttles. Lots of reactions, get our H's. And this is also a place where we will make some CO2 as well. So put some CO2s coming off of here. I don't know exactly where, but you can see where it is if you actually go to the book and look at that diagram. But I'm not too worried about it, as long as you get the main idea. So Krebs cycle, pretty easy with what we're looking at. Just rearrange our molecules until we get enough H's and take those to our grand finale, which is where we're at. The last step, step 4. Any questions on Krebs cycle before we move to this last step? Anybody need a H out there for some money in the bank? I saw a yawn right over there. Woo. Get some ATP made. All right, step 4. You're going to have to do a little drawing here. Step 4 is called electron transport chain, or ETC for short. Electron transport chain, or ETC for short. Draw a beautiful membrane. Now this is where my art skills from high school really start to pay off. I got through college without taking any art classes except theater, so you can really tell that I didn't need it because I'm so good. We're not. All right, so that's a cell membrane. Well, not a cell membrane, it's a mitochondria. It's the inner membrane of the mitochondria. Like we're deep inside the mitochondria now. This is like the secret society place to go where you want to make ATP or get phosphorylated. Then, as I mentioned back in the last chapter, in order to do this, you need a metabolic pathway. You need a chain of enzymes. So because I have such great art skills, I'm going to portray those enzymes by making circles. Look at that. Remember, one of those enzyme complexes in there is the one that cyanide poison inhibits. One of them. From my drawing, you can't tell which one, but that's okay. All right, I did 4 circles. Those portray the enzyme complexes. Then I'm going to make a very special one on the end. And yeah, like I said, that's where my artwork really pays off. The special enzyme on the end is called ATP synthase. In fact, you are going to build a model of ATP synthase. My model today just kind of looks like it's a funnel that stuff can travel down, but your model is going to look much better tomorrow, right? Yeah, this is a very special enzyme. It's the world's smallest turbine, can spin just like a water wheel, and when it does, it has kinetic energy to phosphorylate ADP. However, we have to have something to make it spin. Well, it's a good thing that we have NADs coming. Here they come, NADHs. And here comes some FADH2s. So these H's have been gathered up along the way. We finally have shuttled or delivered them to the proper enzyme complex. NADH delivers there. FADH2 I'm going to have delivered here. They're going to give up their H's. In this case, we've got a couple H's. And of course, when they give them up, then they become oxidized again, right? They were reduced. Now they're oxidized. And they're going to be able to go back and go get more H's, more electrons, and then do this all over again. Remember, H's are so important. This is what we're going to use to spin ATP synthase. Also, don't forget, we can't do any of this without oxygen. Oxygen's waiting in the wings. Because our last step is going to be called oxidative, meaning we must have oxygen present, phosphorylation. That's our last step. So oxygen waiting in the wings, waiting to do that. But first we got to make ATP synthase spin. Oh, don't forget we also have, here's a P waiting, nice and patiently. Here's an ADP waiting, nice and patiently. It's waiting. But we need some energy to put them on there. All right. So now we've kind of set what's going on. So now we're going to pump our protons up here. So make an area up here where we have pumped protons. Remember, protons come from hydrogen. Make an area that's very concentrated with protons. Those enzymes can pump those up there. Meanwhile, the electrons that we get from all of this, we're going to pump this way. We're going to pump those down here. Okay. Didn't I tell you this is what's going to happen in the end? I did this the other day. Made pluses on one side, minuses on another side. I've separated them. I've created a concentration gradient where there's a lot of pluses up there, and they want to go down their concentration gradient, but there's only one place they can do this. Plus we've got negatives down here. They'd like to reunite those negatives down there. Opposites attract. The only way for them to get back down here is to go through ATP synthase, just like a water wheel. That's what they do. They will diffuse down there. When they do, ATP synthase spins. Alright, so I did a little side note there so when I look at my notes later, I'll know that ATP synthase spins as the protons rush down. That's called chemiosmosis. Like if water rushes down its concentration gradient, it's called osmosis. But since we're talking about a chemical here, a proton, then it's called chemiosmosis. So it's the same idea, down the concentration gradient. Only one spot, it's through ATP synthase. ATP synthase spins, it gets kinetic energy. Finally, I can take my P and put it onto ADP to make ATP, which was our goal all along. All those steps, isn't that amazing? All those steps just to get to that point. Meanwhile, remember we had some waste along the way. We got rid of carbon dioxide, C and O. However, we have some waste now. We have pluses and minuses. We have hydrogens. We have our protons reuniting with their electrons. We have hydrogens left over. We can't just let them sit there. That's not good. So we have one more waste to make. Aha! This is another reason besides oxygen attracting the electrons towards it because it's electronegative. The H's, once they recombine here, plus and a minus makes an H. It'll also join with oxygen. Actually, I'll need 2 hydrogens and an oxygen, and I will make H2O. So there's a waste. That's where we get the other part of that equation. We got carbon dioxide, we got ATP, and we got water. If everything was pretty efficient, we could get up to 38 ATP from one glucose molecule. Keeping track, however, that we did have to spend some along the way. It's not always 100% efficient, unfortunately. But up to 38, and that's what makes most of our ATP in our bodies. We don't want to do it that bad way that I talked about earlier. We want to do it this way. All right, any questions on this? Like I said, this is kind of abstract. There's a lot of steps to get to one thing in the end, I know. But we're doing it now, and that means later on in the year, you'll have this done with. Yes. I know. Time flies when you're making ATP. All right, as I said, tomorrow we are going to do some model building of ATP synthase tomorrow. We have a lab coming up as well and some other case study work, and looking like tests next week sometime. Not anymore this week.